One of my patients injected a peptide for several months. He had been promised better recovery and better health, but nothing seemed to be happening. Eventually, he sent an unused vial from the same supply to a laboratory. The result? No detectable active peptide, only what appeared to be filler material. He had apparently spent months injecting pharmaceutical fairy dust. But then I found another case that makes this story much more complicated. In Australia, an accredited laboratory tested a black market vial labeled as containing 10 milligrams of Retatrutide. It reportedly contained Retatrutide, but almost 19 milligrams, nearly double the dose on the label. So the underground peptide market appears to offer a fascinating range of possibilities. Nothing, something, or twice as much something as you ordered. But if these products are so uncertain, why are thousands of intelligent people, including doctors, athletes, celebrities, and Silicon Valley entrepreneurs, using them? Why do so many people insist that BPC 157 healed an injury, Selank reduced their anxiety, or MOTS-c gave them their energy back? Are they early adopters of tomorrow's medicine? Are they experiencing a mass market placebo effect? Or are we watching real science, genuine benefits, and very enthusiastic marketing collide before the clinical trials have caught up? I'm Dr. Low Chai Ling, and this is Chai Files. Today, we're investigating the peptide boom, not to prosecute it, not to promote it, but to answer one question. Do these peptides actually work? Part 1. Peptides are already real medicine. Let us clear up one misconception immediately. Peptides are not inherently fringe medicine. Peptides are short chains of amino acids. Our bodies use them as biological messages, controlling processes such as appetite, metabolism, hormones, inflammation, and tissue repair. Insulin is a peptide hormone. Semaglutide and tirzepatide are peptide-based medicines. Tesamorelin is a peptide medicine. Retatrutide is an investigational peptide producing substantial weight loss in clinical trials. So saying, I do not believe in peptides is rather like saying, I do not believe in tablets. The meaningful questions are which peptide, for which condition, at what dose, through which route, supported by what evidence, and manufactured by whom. Because peptide describes a chemical family, it is not a certificate of effectiveness, it is also not a certificate of nonsense. Part 2. Where did this craze come from? The peptide movement is often associated with Silicon Valley, but Silicon Valley did not invent it. These molecules came from completely different scientific traditions. BBC 157 emerged from Croatian research into gastric protection and tissue injury. Semax and Selank were developed through Soviet and Russian peptide programs investigating neurological function, stress, and anxiety. MOTS-c was discovered by academic researchers examining signals produced by mitochondria. GHK-Cu came from research into naturally occurring copper-binding peptides, wound healing, and skin remodeling. Tesamorelin and Retatrutide came through conventional pharmaceutical research. What Silicon Valley contributed was not the molecules, it contributed a mindset. If the body has a biological pathway, perhaps we can optimize it. That mindset merged with bodybuilding, sports recovery, anti-aging medicine, Russian nootropics, longevity clinics, and eventually social media. Then Ozempic and Mounjaro changed public perception. Their success proved that a peptide medicine could transform appetite, weight, and metabolic health. People naturally began asking, what other peptides are out there? Joe Rogan and other major podcasters discussed recovery peptides. Athletes shared their experiences. Longevity influencers created protocols. Clinics started offering peptide stacks. And overseas manufacturers realized that a growing number of consumers were willing to inject a molecule long before mainstream medicine was willing to prescribe it. That is how unrelated research compounds became one enormous category called peptides. Scientifically, that grouping makes very little sense. Commercially, it was genius. Part 3. Can so many people be wrong? This is the question that makes the peptide debate interesting. If these products do nothing, why do so many people continue using them? When I examined peptide forums, I did not find one single story. I found 3. The first group says, "This changed my life." The second says, I think it helped, but I was also doing other things, so I cannot be certain. And a third says, I injected, waited, and nothing happened. That is not what a simple scam looks like. It is what we might expect when several factors exist simultaneously. Some molecules may have genuine effects, some people may respond better than others, some conditions naturally improve. Many users combine peptides with physiotherapy, diet, or other drugs. Product quality varies, and expectations influence subjective symptoms. Anecdotes are not clinical trials, but anecdotes are not worthless. In medicine, repeated patient observations often generate the questions that clinical trials later answer. The problem is that forums cannot provide a denominator. We see the person who posts, "My shoulder is finally better." We do not see everyone who tried the same peptide, noticed nothing, and quietly moved on with their lives. Therefore, user reports can review a signal, they cannot reliably tell us the success rate. With that in mind, let us examine the peptides people are actually taking. The recovery peptides. Part 4, BPC-157, the Wolverine peptide. BPC-157 is the celebrity of the experimental peptide world. Its story traces back to Croatian research into cytoprotection, the biological processes that protect tissues from injury. Researchers identified a 15 amino acid sequence associated with what they called body protection compound. In animal studies, the Croatian research program reported effects involving tendons, ligaments, muscles, nerves, blood vessels, and the gastrointestinal tract. You can immediately see the attraction. Imagine that you have lived with tendon pain for a year, you have tried rest, physiotherapy, and anti-inflammatory medication. Then you find research suggesting that a small peptide may support tissue repair. Even if the human evidence is incomplete, the temptation is understandable. What do users say? Some describe striking improvement in elbow, shoulder, knee, or back pain. One forum contributor reported that his elbow tendonitis improved while using PPC-157 and undergoing physiotherapy. Another said knee and elbow discomfort felt better within days. One person started it for a tendon problem and reported little improvement in the tendon, but said his long-standing reflux unexpectedly improved. Other people say their symptoms return when the peptide is stopped, and some say they experience no improvement at all. The adverse reports are equally varied. Some users describe headaches, palpitations, increased anxiety, or gastrointestinal symptoms. A small number report emotional flatness or loss of pleasure, which they described as anhedonia. But forum reports cannot establish whether BPC-157 caused those symptoms, particularly when users are combining medications, supplements, and multiple peptides from uncertain sources. What does the formal human research tell us? Very little. One frequently cited pilot involved only 12 women with interstitial cystitis. BPC-157 was administered directly into the bladder. It was small and uncontrolled, without a placebo comparison. It cannot tell us whether injected BPC-157 heals human tendons, but here's the fair conclusion. The evidence does not prove that BPC-157 works for sports injuries. It also does not prove that the many people reporting improvement experience nothing pharmacological. Several possibilities remain open. BPC-157 may accelerate repair, it may influence inflammation or pain without reconstructing the tissue. It may benefit only particular injuries or particular patients, or much of the observed improvement may come from rehabilitation and natural recovery. That is not a verdict. That is a very worthwhile research question. Part 5, TB500, the other half of the Wolverine stack. Now, TB500 is frequently combined with BPC-157. Its origin is connected to thymosin beta-4, a naturally occurring 43 amino acid peptide involved in cell movement and wound biology. TB500, however, is generally described as a much smaller 7 amino acid fragment. That distinction matters. A fragment will retain an important biological effect. It may also behave differently from the full molecule. A chapter can contain the most important sentence in a book, but it is still not the complete book. Users often describe the BPC-157 and TB500 combination as improving recovery, range of movement, muscle soreness, and persistent tendon discomfort. But because the two are frequently taken together, we have no idea which peptide contributed what. A user may also be resting, doing physiotherapy, improving protein intake, and gradually returning to exercise. If recovery occurs, the entire program may deserve credit, but the peptide's individual contribution remains uncertain. The FDA found no human clinical evidence demonstrating that TB500 itself improves wound healing. That sounds discouraging, but it does not settle the biological question. It tells us the consumer experiment has traveled much further than the clinical one. And that gap between widespread use and minimal formal data is the defining feature of the peptide movement. The optimization peptides, part 6. MOTS-c, the mitochondrial peptide. MOTS-c has one of the most compelling scientific stories. It's a mitochondrial-derived peptide discovered in 2015 by researchers at the University of Southern California. Mitochondria are often called the batteries of our cells, but they do more than just produce energy. They also send signals affecting metabolism and cellular stress. Researchers discovered that mitochondrial DNA encoded small signaling peptides, including MOTS-c. In animal models, MOTS-c influenced insulin sensitivity, metabolic homeostasis, and physical performance. For the longevity world, this was irresistible. A peptide connected to mitochondria, exercise, aging, and metabolism sounds almost custom designed for biohackers. But what do users feel? Interestingly, many do not describe a caffeine-like rush. They describe being less fatigued during exercise. One forum user said MOTS-c did not make him feel superhuman, but seemed to improve energy and brain fog. Another reported being able to complete workouts without feeling as exhausted, but without the shakiness of a conventional pre-workout stimulant. Others report no noticeable effect. Some describe temporary itching or irritation after injections. There is also a small human physiology study in which exercise increased the body's own circulating MOTS-c. That supports the idea that MOTS-c participates in human exercise biology. But your body producing more of something during exercise does not prove that injecting it reproduces the benefits of exercise. Your body temperature rises during exercise too. We do not inject fever. Still, MOTS-c should not be dismissed as an imaginary internet molecule. It is an interesting biological signal with encouraging animal data and preliminary human relevance. The unanswered question is whether it can become a useful drug. Part 7. Semax and Selank, the Russian brain peptides. Semax and Selank existed before biohacking became a fashionable word. They were not invented by a wellness clinic or an online peptide seller. Both emerged from Soviet and Russian peptide research programs associated with the Institute of Molecular Genetics of the Russian Academy of Sciences. Semax is a synthetic 7-amino acid peptide related to a fragment of ACTH, a hormone involved in the body's stress response system. Researchers modified the fragment in an attempt to preserve its neurological effects without retaining the hormonal activity of full-length ACTH. Semax was subsequently developed primarily for neurological applications and is registered in Russia as an intranasal medicine. Selank is another 7-amino acid peptide. It was derived from trypsin, a naturally occurring peptide associated with immune regulation, and was developed principally as an anti-anxiety treatment. These are not peptides supported only by mice and internet testimonials. There is genuine Russian human research into both of them. What did the Semax studies find? In a 1997 Russian clinical study, researchers evaluated Semax in 30 patients during the acute stage of ischemic stroke and compared them with 80 patients receiving conventional treatment. The authors reported that adding Semax appeared to accelerate the recovery of certain neurological functions, particularly motor deficits. That is an important clinical signal. However, the index report does not describe the type of modern randomization, placebo control, and blinding that would allow us to confidently exclude bias. A later study published in 2018 followed 110 patients undergoing rehabilitation after ischemic stroke. Patients receiving Semax reportedly experienced increases in plasma BDNF. that's brain-derived neurotrophic factor, and greater improvement on the Barthel Index, which measures a person's ability to perform everyday activities independently. Again, that is not nothing. It suggests that Semax may have biological and functional effects in the human stroke recovery, but the available report does not provide enough methodological detail to determine how patients were allocated, whether assessors were blinded, or how completely adverse events were captured. Cmax has also been studied in healthy volunteers using functional brain imaging. In one small placebo comparison study involving 24 participants, intranasal Cmax was associated with changes in the brain's resting state default mode network. A separate study examining Semax, Selank, and placebo in 52 healthy participants found changes in functional connectivity involving the amygdala and temporal regions of the brain. These studies demonstrate that the peptides can produce measurable effects on human brain activity, but a change on an fMRI scan is not automatically proof that someone thinks more clearly, remembers more effectively, or recovers better from a neurological illness. It establishes biological activity, not necessarily clinical benefit. What did the Selank studies find? Selank has direct human anxiety research. A 2008 Russian randomized controlled study included 62 patients with generalized anxiety disorder or neurasthenia. 30 patients received Selank and 32 received mirtazapine, a benzodiazepine class anti-anxiety medication. The investigators assessed participants using established psychiatric scales, including the Hamilton Anxiety Rating Scale, the Zong Scale, and the Clinical Global Impression Scale. They reported that Selank's anti-anxiety effects were similar to those of mirtazapine. Selank was also associated with anti-fatigue and mildly stimulating effects. That is meaningful evidence. It is considerably more than an anecdote or an experiment in rodents, but it was still a small study. The comparison was against an active medication rather than placebo, and the publicly available report does not establish clearly that the trial was double-blind. The findings have also not been confirmed through large independent international trials. Later, Russian researchers compared Selank with other anti-anxiety strategies and explored its use alongside medicines such as phenazepam. Those reports generally described reductions in anxiety with less sedation, but they come from the same relatively concentrated research ecosystem. That does not make them false. It does mean that independent replication would greatly increase our confidence. What do users say? On Western forums, the reputations of the two peptides are surprisingly specific. Semax is usually described as the focus peptide. Users report clearer thinking, reduced brain fog, improved concentration, more motivation, and sometimes better verbal fluency. Selank is described as the calm peptide. Users report a reduced background anxiety, less overthinking, a calmer stress response, and relaxation without feeling heavily sedated. Some combine them and describe the result as focus without tension. One user said the combination made him calmer and less likely to lose his temper. Another described finding words more easily in conversation. Others say the effects are extremely subtle, and several users report absolutely nothing. The consistency is interesting. People often describe effects that match the purpose for what each peptide was originally developed. That could represent a genuine pharmacological signal. It could also be influenced by expectation because users already know what each peptide is supposed to do. Those explanations are not mutually exclusive. What did the FDA say? The FDA's 2026 review concerned Semax, not Selank. It evaluated Semax for cerebral ischemia, migraine, and trigeminal neuralgia. The FDA did not say that Semax had never been studied or that it had no biological activity. Its scientists concluded that the available evidence was insufficient to establish effectiveness for the specific uses under review. They also identified gaps involving human pharmacokinetics, long-term safety, immunogenicity, product stability, nasal delivery consistency, and uncertainty over whether particular studies use Semax-free base or Semax acetate. The Independent Advisory Committee, nevertheless, voted 8-5 with one abstention to recommend Semax for possible inclusion on the Section 503A bulks list. That was not FDA approval, it was a recommendation concerning a possible pathway for patient-specific compounding. Selank was not included in that July 2026 review. Therefore, claims that the FDA recently cleared Semax and Selank are incorrect. Where do Semax and Selank belong on the evidence spectrum? They should sit above peptides supported almost entirely by animal studies. They have genuine pharmaceuticals histories, human clinical research, and evidence of measurable biological activity, but they remain below an FDA-approved medicine such as Tesamorelin or an extensively studied investigational drug such as Retatrutide. The most accurate in conclusion is not there is no evidence. It is there is genuine and encouraging human evidence, but not yet large enough independently replicated research to know how reliable the benefits are, who is most likely to respond, or how safe long-term use will be. Perhaps the correct question is no longer, do Semax and Selank do anything? There is evidence that they do. The more difficult question is whether those biological and clinical signals are strong, reproducible, and safe enough to support much wider use. Part 8. GHK-Cu, the beauty peptide. GHK-Cu is a naturally occurring copper-binding tripeptide associated with tissue remodeling and wound biology. Unlike many experimental peptides, it has a long-standing history in topical skincare research. Laboratory studies and small human studies provide a reasonable scientific basis for investigating topical GHK-Cu for skin repair and aging. People using it topically report better skin texture, less redness, improved recovery, and sometimes thicker-looking hair. Injectable GHK-Cu is a different proposition. Some users say they notice broader improvements in skin quality after several weeks. Others report painful stinging, redness, itching, or swelling. I've also seen patients injecting GHK-Cu obtained from overseas sellers. One patient used it for several months without seeing any improvement. When the product was sent for laboratory testing, no active GHK-Cu was detected. That does not invalidate GHK-Cu as a molecule. It invalidates our assumption that a label proves what is inside a vial. Topical evidence also cannot automatically validate injections. The route changes exposure, dosing, and risk. So GHK-Cu represents both sides of the peptide story perfectly. A real molecule with credible biological relevance and a consumer market making claims far beyond what has been established. When peptides become conventional medicine. Part 9. Tesamorelin, proof that peptide therapy is real. Tesamorelin demonstrates what happens when a peptide crosses the clinical finish line. It stimulates the growth hormone pathway and has been evaluated in substantial human trials. Tesamorelin is FDA approved to reduce excessive abdominal fat in adults with HIV-associated lipodystrophy. Pooled phase 3 data included more than 800 participants and demonstrated reductions in visceral abdominal fat. That is not an anecdote, it is measurable clinical evidence in a defined population. Online, people use Tesamorelin outside that approved population and report changes in waist size, body composition, sleep, and recovery. Some track DEXA scans, IGF-1, and waist measurements. Others use the highly sophisticated scientific instrument known as How My Trousers Fit. Tesamorelin proves that a peptide can produce a genuine and clinically meaningful benefit, but it also teaches us about indication. Evidence in HIV-associated lipodystrophy does not automatically prove that the same risk-benefit balance in healthy adults pursuing fat loss or longevity. Tesamorelin affects the growth hormone and IGF pathway that requires clinical judgment and monitoring. It is real medicine. It is not a causal wellness vitamin. Part 10. Retatrutide, the peptide that changes the argument. Retatrutide is where the story becomes even more interesting. It is Eli Lilly's investigational triple receptor agonist designed to activate GIP, GLP-1, and glucagon receptors. In a randomized phase 2 trial involving 338 adults with obesity, participants receiving the highest dose lost an average of 24.2% of their body weight over 48 weeks. The placebo group lost approximately 2.1%, so those results are extraordinary. Retatrutide demonstrates that the molecule can be investigational, not yet approved, and still have powerful human evidence. But the black market began selling Reta while Lilly was still conducting its clinical development program. Retatrutide was not discovered by biohackers and later adopted by Lilly. It was Lilly's proprietary molecule. Its structure became publicly accessible through patents and scientific publications. Outside manufacturers could therefore attempt to synthesize it. But a published blueprint is not a finished pharmaceutical product. To reproduce retatrutide correctly, a manufacturer must assemble the correct amino acid sequence, place the modified amino acids properly, attach the fatty acid side chain at the intended position, remove truncated or incorrectly assembled material, control degradation and aggregation, achieve consistent potency, and manufacture the final injection under sterile conditions. This brings us back to that Australian sample. The vial was labeled as containing 10 milligrams. Laboratory testing reportedly identified approximately 19 milligrams of retatrutide. So at least some underground products appear to contain active material consistent with the claimed drug. The problem is not simply that black market sells nothing. It may also sell something very powerful in an unpredictable quantity. A vial containing no active drug wastes money and creates injection risks. A vial containing twice the expected amount may create a medical emergency. Fairy dust is bad. Fairy dust with unexpectedly potent receptor activity may be worse. Part 11. Why haven't all these peptides been properly studied? Peptide advocates raise an important point. What if some older peptides have not received major trials because nobody can make enough money from them? Clinical development is extraordinarily expensive. If a peptide sequence has been publicly known for decades and cannot be strongly protected, a pharmaceutical company may hesitate to spend hundreds of millions proving that it works. Why finance the evidence if competitors can later copy the molecule and benefit from your investment? This is a genuine weakness in the market-driven medicine. Potentially useful treatments may remain under-investigated because they are commercially unattractive. But this distinction is essential. Commercial neglect explains why evidence may be absent. It does not tell us what the missing evidence would show. The trial could be positive, it could be negative, it could reveal a small benefit in one very specific population. Patent protection is also not always impossible. Companies may protect modified sequences, longer acting analogs, formulations, delivery systems, manufacturing methods, or particular clinical uses. Regulatory exclusivity is not separate from patents and can also reward new drug development. The fair conclusion is not pharmaceuticals ignored it, therefore it must work. It is some peptides may be under-investigated for economic reasons that should make us more willing to fund independent research, not more willing to pretend the research has already happened. Otherwise, profitable molecules receive phase 3 trials, while unprofitable ones are studied through Reddit comments. Part 12. What the FDA vote really meant. In July 2026, the FDA's Pharmacy Compounding Advisory Committee considered 7 peptide families: BPC 157, KPV, DB 500, MOTS-c, amideltide, or DCIP, Epitalon, Semax. The committee recommended 6 for possible inclusion on the Section 503A bulks list. Amideltide did not receive a favorable recommendation, but the committee's vote was non-binding. This was not FDA drug approval. It did not establish that the peptides were safe and effective. It did not legalize every product sold online. If the FDA eventually adds a substance to the 503 bulks list, certain state-licensed pharmacies may be permitted to compound it for an individually identified patient under defined conditions. Supporters argue that this could improve product identity, sterility, traceability, medical supervision, adverse events reporting, and batch accountability. Their position is that their patients are already using these substances. A regulated compounding route may therefore be safer than forcing them to its anonymous overseas seller. That is a reasonable harm reduction argument. FDA scientists took a more cautious position. They emphasized limited evidence, unresolved safety questions, and manufacturing concerns. That position is also reasonable. The disagreement was not simply between people who believe in peptides and people who do not. It was a disagreement about how medicine should respond when widespread use arrives before adequate evidence. Part 13. What my patients tell me. My patients' experiences reflect what we see online. Some report meaningful benefits. They describe better recovery, more energy, less discomfort, or improved skin. I do not tell them that their experience is imaginary because a phase 3 trial does not exist. Others say they spent months taking peptides without noticing any improvement. Some develop itching, redness, or swelling around injection sites. Those reactions could come from the peptide, but they could also result from an excipient, an impurity, peptide aggregation, contamination, improper storage, injection technique, or an immune reaction. When we do not know what is inside the vial, we cannot confidently attribute either the benefit or the side effect to the label. That is why the tested vial from my patient matters. The fact that it contained no detectable active peptide does not prove that all unregulated peptides are fake. The double-strength Retatrutide sample proves that they are not necessarily fake. Together, the two cases expose the deeper problem. The product may contain too little, too much, the wrong substance, or the correct substance with unknown impurities. Before discussing pharmacology, we must establish identity. Part 14. Could it all be placebo? No. The entire peptide market cannot credibly be explained as placebo. Some peptides have established pharmacological effects. Tesamorelin alters visceral fat in controlled trials. Retatrutide produces substantial weight loss. Growth hormone releasing peptides can change measurable hormones even when their long-term clinical benefits remain uncertain. For experimental peptides, expectation probably contributes to some reported improvements, particularly subjective outcomes such as pain, energy, anxiety, and concentration. But placebo is not lying. Expectations can genuinely alter symptom perception, stress responses, behavior, and adherence. The more realistic explanation is that several effects are mixed together: genuine pharmacology, natural recovery, expectation, concurrent treatments, lifestyle changes, selection bias, product inconsistency, and marketing. The proportions differ between peptides and between people. That is why the correct answer is not they work or they do not. It is some have real effects. We do not yet know how reliably those effects occur, who benefits most, or whether the benefits justifies the risk. Part 15, my peptide evidence spectrum. Rather than dividing peptides into good and bad, I place them on an evidence spectrum. At one end are approved peptide medicines such as Tesamorelin. They have defined manufacturing, controlled human trials, and a specific approved indication. Next are the investigational pharmaceutical molecules such as Retatrutide. They may have strong clinical evidence, but development and regulatory evaluation are still underway. Then we have Semax and Selank. They have genuine foreign clinical use, human clinical studies, and evidence of biological activity, but still require larger independent replication. Next are molecules such as BPC-157 and MOTS-c. They have compelling biology, encouraging preclinical research, and a strong user signal, but very limited reliable clinical evidence. GHK-Cu occupies two positions simultaneously. Topical GHK-Cu has plausible skin signs and some human evidence. Injectable GHK-Cu remains much less established. TB500 sits closer to the speculative end because popular claims rely heavily on related thymosin beta-4 biology and uncontrolled user experience. This spectrum can change. A peptide that is experimental today may become established tomorrow. Another may fail when properly tested. That is how science progresses. So, conclusion, do these peptides actually work? Some definitely do, some plausibly might. Some may work only for narrow conditions or particular patients, and some may ultimately prove to be expensive dreams. I do not believe that everyone using experimental peptides is foolish. People are responding to real problems: chronic injuries, fatigue, anxiety, aging, poor recovery, and dissatisfaction with treatments that often feel slow or incomplete. Some users may be early adopters of tomorrow's medicine. Some may be improving because of rehabilitation, lifestyle changes, or a natural recovery. Some may be experiencing placebo effects, and some may be taking a genuinely active molecule that has not yet received the research it deserves. Most likely, all these stories are happening at once. The answer is not blind enthusiasm. It is also not mockery. We need to listen to patients, investigate recurring signals, verify what products contain, and conduct better trials. Because we do not know yet is not the end of scientific conversation. It is the beginning. Perhaps the peptide users are early adopters of tomorrow's medicine. Perhaps some are buying beautifully marketed whole. The only way to separate those possibilities is better evidence and knowing what is actually inside the vial. If you have taken a peptide, I want to hear from you. But please tell me 3 specific things. What did you take? What objectively changed? And was the product independently tested? Your experience cannot replace a clinical trial, but it may help us ask a better scientific question. I'm Dr. Low Chai Ling, and this is Chai Files. Subscribe if you want honest, evidence-based discussion about beauty, medicine, and longevity without pretending that uncertainty is the same as ignorance. Stay curious, stay honest, and please remember, research use only is not a medical qualification.